So in the past few sections, I've talked about how relatively long-term synaptic weight changes are managed at various levels of description detail. But there are a few other processes that are important for higher level understanding that I'll now describe. There are a range of mechanisms that can increase synaptic weights for relatively short periods after inputs have been received. And one mechanism, called augmentation, lasts for periods of the order of seconds. If input is provided to a synapse, the augmentation mechanism results in an increase in synaptic weight which decays over a period of about 10 seconds. And the detailed processes that support augmentation are different from those supporting longer-term synaptic weight increases. In particular, they operate in the presynaptic bouton to increase the amount of neurotransmitter released in response to one action potential. If we go back to this diagram of a synapse, there are lots of vesicles containing neurotransmitter. Vesicles fuse with the membrane and release their contents into the synaptic cleft in response to an incoming action potential, and the neurotransmitter diffuses across the gap to open sodium channels in the target neuron. When an action potential arrives, it opens voltage-gated calcium channels in the membrane of the presynaptic bouton. Calcium ions diffuse in through the open channels, triggering the opening of vesicles docked on the membrane. The neurotransmitter then opens sodium channels, injecting postsynaptic potential into the target neuron. The voltage-gated calcium channels close, and the calcium ions inside the presynaptic neuron are removed. However, this removal of calcium ions takes time, and some ions may still be there a few seconds after the action potential. If another action potential arrives in this period, a somewhat higher concentration of calcium ions may result. The higher concentration can lead to a larger number of vesicles releasing their contents into the postsynaptic cleft. As a result, more sodium ion channels open, and the postsynaptic potential injection is a bit larger. And as I mentioned, this effect can last for a number of seconds. A second, rather different chemical process I'd like to touch on occurs in inhibitory GABAergic synapses. I mentioned earlier that although GABA receptors are generally inhibitive, they can under some circumstances be excitatory, depending on the relative timing of glutamatergic and GABAergic inputs. In particular, if the GABA input arrives after or just before the glutamatergic input, it's inhibitory. But if it arrives well before the glutamatergic input, it can slightly reinforce the effect of the slightly later excitatory input. If they're widely separated, there's no interaction. It's one particular type of GABA receptor, the GABA-A receptor, that's responsible for this effect. Now remember that inhibition can be the result of the opening of either chloride or potassium channels. There's more chloride ions outside, and when they flow in, they carry negative charge, while because there's more potassium ions inside, when a channel opens, some potassium ions flow out, and positive charge going out is equivalent to negative charge coming in. The GABA-B receptor is a G protein receptor and inhibitory on a relatively longer time scale because it affects potassium channels. The GABA-A receptor is an ion channel, so it operates faster and is sometimes slightly excitatory. To understand how this can be, you first need to know that an open GABA-A ion channel can pass chloride and bicarbonate ions. The concentration of chloride ions is higher outside the membrane, but the concentration of bicarbonate ions is usually greater inside the neuron. So a flow of chloride ions will increase or hyperpolarize the membrane potential and therefore inhibit the neuron, but a flow of bicarbonate ions will depolarize the neuron. And the degree to which the channel passes the different types of ions depends on the current membrane potential. If the membrane's at resting potential, then bicarbonate ions flow more easily, and so the membrane is depolarized. On the other hand, if the membrane is already slightly depolarized, flow of hyperpolarizing chloride ions is favored. Now, the bicarbonate depolarization is only slight. Significant depolarization will simply change the channel and allow chloride ions to pass, but this slight depolarization can reinforce the effect of a later glutamate action potential. So, if the synapses are close together, the GABA synapse will inhibit or reinforce the effect of the glutamate synapse depending on the relative arrival times of the action potentials. Now, some GABAergic neurons play an important role in forcing a synchronization onto targeted pyramidal neurons. Under normal circumstances, the inputs to a pyramidal neuron are uncoordinated in time, and the outputs from the targeted neuron will therefore also be uncoordinated. However, if the neuron's also targeted by GABA spikes at a regular frequency, the probability of an output is reduced close to a GABA spike, but may be slightly increased when offset from a spike. So there'll be a tendency for the targeted neuron to produce spikes at the same frequency, but with a phase difference from the GABA spikes. Remember, we talked earlier about an integration window. Incoming spikes need to be within about 8 milliseconds of each other to be able to reinforce. If GABA spikes at a particular frequency are applied to a population of neurons that provides input, say, to a second population, the outputs from the first population will tend to be squeezed closer together in time, rather than just being reduced in number. So spikes will tend to occur clustered at points in time a little bit offset from the GABA spikes. So on average, multiple spikes are more likely to occur within the same integration window, so outputs from the second population are more likely. In other words, if you have two populations of neurons, one providing outputs to the other, applying a GABA modulation signal to one population of neurons makes it more likely that the other population targeted by the first will produce outputs. If speed is of the essence, direct electrical synapses from one neuron to another exist. There can be a direct flow of ions from one neuron to another. These synapses, often called gap junctions, allow very fast interactions between neurons, for example in synchronizing the activity of interneurons. Each neurotransmitter has different detailed properties that result from the properties of its receptors. And I'll close this section by giving a bit more detail about the neurotransmitter dopamine. Dopamine has a number of different receptors, which can be organized into two classes. And sometimes dopamine can leak away from synapses and be present at some level throughout the extracellular environment. In this situation, some dopamine receptors may be located in the membrane wall well away from any synapse and exert a general effect on the neuron. Then, in addition, dopamine can have opposite effects on different neurons depending on the type of receptors and where they're located. These different types of receptor play an important role on synaptic weight changes, which complicates the relatively simple picture I drew earlier, because depending on the relative activation of different receptors, synaptic weights can increase or decrease. And one other dopamine phenomenon is that some dopaminergic neurons can either fire constantly at about 4 spikes per second, or occasionally produce bursts of spikes at a rate of about 20 spikes per second within the burst. And as we'll discuss later, these different firing modes by the same neurons perform different functional roles. So now at this point, we're going to go on to begin the description of the anatomy of various major structures, now that we've covered the underlying neuron physiology.